In this video, we're going to practice DNA transcription and translation. We're also going to do a little replication. So over here, you see that I have a codon chart. I realize that it's probably too small for you to view in the video, but you do have a codon chart in the PowerPoint that you can reference. We'll talk about how it works later. I want to start with DNA replication. You already saw a video on the molecular level of DNA replication. Now we're just going to look at the nucleotides themselves, so those base pairings, right? Our DNA is 5'. We'll just put some random any old nucleotides will do. That's probably plenty. So we have 5' to 3'. In replication, we're making a second strand. So this is what's happening in that S phase of interphase, remember? Also remember that if one strand is running in this direction, then the other strand is running in the opposite direction because they're anti-parallel. So that means that 5' would start over here and go this way, and 3' is over there. So all we have to do to fill these in is match which nucleotides pair with what. G always pairs with C, and A always pairs with T, which means for a T, it's an A, for a C, it's a G. We can fill the rest in. That's all you have to do. So you could write down any combinations of G, A, T, and C to practice. You'll also have a practice problem on your genetics assignment. Let's do transcription. Transcription is going from DNA to RNA. Let's get ourselves a strand of DNA going. This time we'll just start with the 3', just for fun. It doesn't really matter what you do. Okay, let's do some random cat, gag, ugh. Let's do one more. AT&T for the stadium, for all our Dallas Cowboy fans. All right. In RNA, remember that there is no T. And so anytime that we thought we were going to write a T, we should instead write a U, because uracil replaces thymine. So this side is 3', which means this end is 5'. T pairs with A. We still have A, so we can write an A. A now pairs with U. So before A paired with T, so we're going to put a U, right? C pairs with G, that stays the same. C pairs with G, A pairs with U, T pairs with A. You get the picture. We can just fill these in. And this end will be 3' because that end was 5', right? So it's the same kind of process. You're just replacing any T's you would have written with U's. The last step is the one that's going to involve our codon chart, and it is translation. So DNA to RNA was easy. They're the same language. They use kind of the same letters, right? Translation is a whole different ballpark because we're moving from RNA to amino acid letters. Protein language is a completely different language. And we're actually going to build our protein from this one we just made, this strand of RNA we just made. And we'll be using the codon chart. The first thing we want to do, though, is break these into codons. So every 3 nucleotides of RNA is a codon. 1 codon, 2 codons, 3 codons, 4, 5 codons. Right? So 3 letters of RNA language will translate into 1 letter of protein or amino acid language. And this is where our chart comes in. So you can kind of see in the smallness that it is that there's these red lines around the outside, and they say U, C, A, and G. So when we look at our codon, if we start with A, we want to find the block that's called A, which is this row right here. All of these start with A. Our second letter is U. The second one is going to be the columns. So we find the column that starts with U, which means it's definitely in here. All of these have a second nucleotide as U. So we had A and U. So that means everything in this one block are AU something. Our last letter is G. So we just check the 4 of them. AUU, AUC, AUA, AUG. This is ours. And it says that it's methionine, or M. We can use the 1-letter code, M. You'll notice this one's green and says start. Every single protein starts with methionine. And that's because on the ribosome, when it's reading that mRNA, it has to know when to bring the first tRNA. It's not just gonna bring it at the very end, because remember we have that 5' cap. And so in order to know where the gene actually starts, it will always start with AUG. So our first one is M. Our next one, the nucleotides are GUA. So we're gonna find that codon on our chart. This group is G, again with U in the first column, and then go down to GUA, and it codes for valine, which is a V. You'll notice, if you're referring to your codon chart, or if you can see this, that all 4 of these code for valine. And that's because sometimes that third one doesn't really matter, that third nucleotide, right? They can all match up, because we have 64 different codons, but only 20 amino acids to work with. So sometimes they'll all be the same, sometimes they'll be different, like those ones. Our next one is CUC. Let's find that codon. CU, I should have chosen less U's. CUC is leucine, so we'll write an L. You don't have to memorize this. I will always give you this chart. You can print the chart and use it on the test. I'm gonna put it on the test questions, so this is not something to memorize, just a tool to use. GGC. G is down here. We look for the column that's G, which is the 4th one. So that means our answer is over here somewhere. GGC is glycine, so that says G on it. We only need the 1-letter code. We don't need the 3-letter code. And our last one is UAA. U is up here. A is the 3rd column. UAA says stop. And so just like methionine told us where to start, there are 3 codons that tell us where to stop. And on that ribosome, when we are translating this mRNA, we have to know when to quit. We don't just want to go to the very end, because we have that long poly A tail on there, and that would leave us just putting a whole bunch of K on the end, and we don't want that. So stop brings a releasing factor instead of a tRNA, and that's the official end of the protein. Now, obviously this isn't long enough to be a real protein. They're like 1,000 long, but you see the process. So on your genetics assignment, you'll have replication, you'll have transcription, you'll have translation, and then you'll have a bunch of Punnett squares to do.